Okay, hello everybody. We're ready to get going. Yep, so thank you everybody for coming. This is our January 2026 meeting of the LA Mycological Society. We meet here on the third Monday of every month from October to June, except February. And that's the first big announcement, which is reminding everybody that our annual mushroom fair is February 15th. It's a Sunday at the LA Arboretum in Arcadia. It'll be the 42nd annual fair. So first of all, I should say that this is an entirely volunteer-run fair. So you're all welcome to volunteer, and that gets you free admission to the fair. Otherwise, admission is run through the LA Arboretum. It does get very crowded, so it's a good way to make sure that you have a spot when we send out this volunteer announcement. So I know we have some announcements in this regard. Vanessa.
Yeah. So like Rudy said, we're recruiting volunteers for the fair. You don't necessarily have to have any specific experience in going to the fair or being a previous fair volunteer. But we have different kind of like job assignments or different things you can do at the fair. Like we'll need volunteer photographers, volunteer sign artists. If you have kitchen experience, we need help in the kitchen, preparing the food that we're going to be selling at the fair. You don't have to have professional experience, but if you like being in the kitchen, that would be something good to volunteer for. If you do want to be a kitchen volunteer, we do ask that you get your ServSafe certification, and then LAMS will reimburse you for the cost for that. Like Rudy said, volunteers get free admission to the fair, and we will need volunteers for the Saturday before the fair and then the day of the fair.
Valentine's Day.
Oh yeah, Valentine's Day. So something romantic. That's what you're feeling. We also need some sign artists. So if you're an artist and you like to paint like graphics or letters, that would be a good volunteer position for you. Another thing, we also make like a candy cow mushroom lemonade for the fair, and it is citrus season in SoCal. So if you have a lemon tree, you can volunteer to donate lemons for the lemonade. Also, we have some art and photo contests going on. So the deadline is going to be ending February 1st. So if you want to submit any art or any of your photography of mushrooms, please send that to the, we have some flyers on the table over here. So you can send it to the email listed on the flyer, and Megan will respond to you. So this is a good way to get your art or photography displayed at the fair, because you will need to bring your piece to the fair. And then tickets to the Arboretum for the day of the fair are going to be available like about 2 weeks before the actual date. So start looking to buy your tickets about 2 weeks ahead of time.
If you're not going to volunteer.
If you're not going to volunteer, yeah. Volunteers get in for free.
And we do get, it's a tremendous turnout that we have, so it will be sold out.
Yeah, it tends to sell out, so. And then also there is limited parking at the fair venue, so try to carpool if you can.
Yeah.
That's it.
Okay, so that brings us to our main presentation today. So I'm, in addition featured presenter each month as well. And this month we're very, very excited to have Dr. Anne-Marie Macara presenting for us today. Anne-Marie is a bioartist, a PhD in neuroscientist, and an educator. And I mean, I certainly wish I could have had a teacher like them when I was in high school. So I'm very, very excited to give the floor to Anne-Marie.
Thank you so much. I'm Anne-Marie. I'm super excited to be here. I remember about 5 years ago when I first joined LAMS, I was sitting in the back over there listening to presenters talk about these really cool, interesting projects, and I was sitting there thinking to myself, wow, I really hope one day that I have something really cool and interesting to present. And so here I am today in an attempt to do just that for you all. And so it's like a real full circle moment for me. And also I just want to name that, you know, in the world there's a lot of sort of awful and horrific things happening here and globally. And I think as an educator, sort of navigating those challenges is particularly difficult. And I think one thing that I try to do is help sort of promote the sense of hope. And one thing that my goal for today is to instill a sense of hope through bringing in mycology, bringing in bioart into the conversation of how we might rethink and rebuild the future of science education for the next generation of students. So with that, let's jump into what I'm going to be talking about today. So first, I'll be talking about science as a creative field, and then we'll explore how fungi have been historically left out of the sort of scientific conversation and also in education as well. And then I'll introduce what bioart is, why it's important, and then share some student examples, and then talk a little bit about how you all might engage in the practice of bioart, either informally at home or in the classroom. So I'd like to just share a little bit about my background and experience. I worked in a research lab studying fruit fly neural development in graduate school. I worked in a sort of a dark closet using this confocal microscope that you see here, imaging hours and hours of fruit fly brains. And so I started inside in the research lab, and then after graduate school, I decided that I wanted to escape the dark closet of research and sort of move into the exciting outdoor world where I became an outdoor educator. And then I moved from that experience into the classroom. And so I share this to let you know that I have a varied perspective of science education, and it's informed me about what science education is missing and how we might shape science education for the future. I just wanted to give you like a little peek into my classroom just so you can get a sense of my vibe. So here is a little like 5-second tour of my classroom. You can see that I have student work all up on the walls. And of course I also sort of surrounding this video, there's a few images that I wanted to highlight, especially for this group here. So of course I've got some mushrooms, some samples. I've got Julie Beeler's mushroom color atlas. And then up here we have the LAMS spore print available for students to like read and explore. And then I also have some mushroom specimens that we've like collected from going out on mushroom forays. So, in contrast to that, when we think about traditional science classroom, we may encounter something that looks like this. So, in a traditional science curriculum, students are often given a textbook or facts where they have to memorize things about science. And this is something that they are consuming rather than actively doing. And so they're often also not given opportunities to perform real experiments. When they are performing experiments, these experiments have known endpoints, and they're either graded that they've got it correct or not correct. And this is not true science. So there's a real disconnect between school science and real, authentic science practice. And so this leads me into my essential question for this talk, which is, what if students discovered that science is as creative a field as art? So to introduce this concept, I'm going to be playing a video that a student animated specifically as an introduction to my advanced biology course. I forgot to mention that I teach 11th and 12th grade students. I teach 2 courses, an advanced biology and biotechnology course, as well as a neuroscience elective. Anyways, so this is from a student. She made this at the end of taking my advanced biology course, and I'm hoping that this helps answer this question, and there may or may not be sound for this. Okay. So I hope this highlights that at least this student has found that science is as creative as art. This shows that science is beautiful, it is exploratory, and it can be actively engaging. So we have, most of us perhaps, have grown up learning about the scientific method as a linear series of steps that has now changed into this model up here, and we call it the scientific process to try to get away from that linear thinking. And as you can see, science truly is a sort of a living process. And we come into this process through curiosity. And from curiosity, we then try to work on our observational skills. We then observe things, we ask questions, those are our research questions, which then translate into developing a testable hypothesis, and that then we can go to create experiments and collect data, and from there we might get like feedback from other scientists or our peers, and that informs scientific communication, like what have we discovered, and then ultimately it might have some benefits and outcomes that help like a society or as a world. And so this is all interconnected, and this is all sort of like feeding off of each other in this sort of dynamic process. Let's step back a little bit and talk about this question of who decides what counts as worthwhile science. So the scientific method was developed and promoted with the start of the Royal Society in London in 1660. And so this Royal Society was a group of philosophers, of naturalists, and other wealthy people. And so they came together with the idea of organizing science, and they controlled funding, who gets funded, they controlled what gets documented, and they controlled what gets taught. So one prominent person that was also part of the Royal Society is Carl Linnaeus, and he was the founder of taxonomy. Taxonomy is the classification of living things. You might recall like domain, kingdom, phylum, class, etc. He was responsible for the binomial naming system of genus and species, and he also classified organisms into two main kingdoms, animals and plants. And one of the reasons that he did this was he was a botanist. He was really interested in plants, and in particular, he was interested in understanding the reproductive structures of organisms. And one thing he couldn't wrap his head around was fungi. And these were like, had open disdain, dislike for fungi. He called them lower plants. He thought that they were chaotic, they were distasteful. And so he put them within the plant kingdom, but they were less than plants. They weren't really respectable to study. And so this informed science for over 250 years. And so this is just the concept that taxonomy, this classification system, isn't just naming organisms, it also determines the legitimacy of studying those organisms. And so for 250 years, his dislike of fungi really reduced people's scientific efforts in classifying and naming and seeing the range of biodiversity in the fungal kingdom. So in 1969, Robert Whittaker came around and he looked at various organisms and found, instead of looking at reproductive structures, he was looking at how these organisms obtained energy. And so he found that there was three main ways organisms did that: through photosynthesis, which was the plant kingdom, through ingestion, which was the animal kingdom, and then through absorption, which he created the new kingdom of fungi. So finally, after 250 years, fungi were, rightfully brought up into the kingdoms of organisms. However, by that point, the damage was already done. In fact, in the 1800s, culturally, there was also this distrust and this dislike, this disdain of fungi. In fact, it was so much so that the mycologist William Hay coined a term for it. He called it fungophobia, which is what we now know as mycophobia. And he described this widespread fear and ridicule of anyone who is interested, even like as a mushroom hobbyist or anyone who is studying mushrooms, that they were to be ridiculed. And so fungi at this time became culturally associated with decay, disease, and death. And this fear of this kingdom was also sort of reinforced by not being discussed or researched in science and education. And this mycophobia and misclassification of fungi had structural impacts that we see today. So there is no standalone mycology departments in the United States. And this has a lot of downstream consequences. So for example, there are very few undergraduate pathways to study mycology. And because there are fewer students, there's fewer labs, there's fewer departments, and there's less funding for mycological studies. And this scarcity then trickles down into the K through 12 education system. Psychedelics have also highly stigmatized the study of fungi. And this was not by accident. This is actually a historical phenomenon that occurred with the 1960s psychedelic movement. So unlike plants, which there are many psychoactive plants, plants have been around for a while. People have been using psychoactive plants for a really long time in like Western society. They've been integrated into the culture. So they are not seen as poisonous or dangerous, rather they're seen as medicinal, as nutritional, and they have health benefits. So they are considered safe. Mushrooms, on the other hand, they have been introduced, psychedelic mushrooms have been introduced into the 20th century abruptly. And because of what that stood for at that time, disorder, misconduct, it got associated even strongly with mycophobia, and it sort of increased and stigmatized fungi at the level of the entire kingdom. And in fact, I have I've encountered this stigma personally as an educator who studies mycology. And I find it very interesting that people have certain judgments of me when I tell them at back to school night that I teach mycology, and they look at me as if I am teaching their students something quite dangerous or uncomfortable. And I really question that, and I push back on that, and I wonder, where is this fear coming from? And it's coming from this moment in history. So it's interesting to think about what feels appropriate to study or teach at the K through 12 level. And there is this double standard in science education. And we can see that when we examine the kingdom of plants and the kingdom of fungi. So for plants, they are, botany is one of the core points of focus in K through 12, life sciences and biology. Plants feel familiar, visible, safe. A lot of schools have gardens. They grow all sorts of plants in. They're central to food, medicine, ecology. If you step back and look at the reality of the plant kingdom, many plants are actually toxic and lethal. There are many plants that are psychoactive. Some are quite dangerous to touch, but they are still widely accepted as legitimate objects of study. Now, if we look at the fungal kingdom, they are rarely centered in K through 12 curricula. They're often framed when they are presented as mold or causing disease, contamination, or death. And they're also frequently avoided due to stigma. So again, if we sort of step back and look at the fungal kingdom as a whole, actually no mushroom in North America is poisonous or dangerous to touch. I actually have another little story. There was a colleague that told me that one of the camp counselors for her second grader was really concerned that the student was interested in picking up mushrooms or even talking about mushrooms. And again, it's just this really interesting moment where it's where is that discomfort or fear coming from? Because fungi play such an important role in ecosystems, in medicine, they have a wide range of applications in biotechnology, and because of this historical bias, there's a vast majority of species that remain undiscovered. Before I talked about bioart as a contemporary thing, I do want to talk about that science and creativity have been around since the emergence of humans. And so cultures around the world have been exploring the natural world in both a scientific and creative way. And some examples of that are these polypore mushroom masks from an indigenous group in Nepal, as well as these really cool wall pockets made by the Tlingit people in Alaska, made from mycelial mats. I believe what they do is they remove the bark from the tree and collect the mycelial mats underneath that. And so these examples highlight and show that knowledge of the world around us, of the natural world, doesn't necessarily exist solely within the realms of Western science, but there are many different ways of knowing and working with natural materials in the world. Let me start. So I think one of really amazing examples of someone who brought in science and creativity and also brought a relational aspect into science is George Washington Carver. So George Washington Carver was born into slavery. Carver is, his last name is from his slave owner. There's also like a little fun fact that Washington was actually a name that he gave himself. He went to college, there was another George Carver there. So he just wrote W to differentiate himself from that George Carver. And when he was asked what the W stood for, he said Washington. And so he, from a very young age, he was interested in the natural world around him. He loved learning about all of the plants and flowers around him. He became known as like the plant doctor in his local community, even at the young age of 12. He was also really interested in art. He painted all the nature that he saw. Again, he was really interested in painting like flowers and other plants. Another interesting thing, he crocheted, he knitted, he did ceramics, he did a lot of creative work, and he brought that in to when he eventually moved into doing a master's in agriculture. He moved in from art to, in college, into science. And he became a botanist. He studied plant pathology, and he also studied mycology. So he found and named several species of fungi. Here's an example of a microfungus that was actually named after him. And what's really cool is he really used the natural materials around him. He went and collected various different types of clay and soil and made paint pigments out of them. And one of his goals with creating paint pigments out of clay was to create a cheaper alternative for his community to paint their houses with, rather than the synthetic paints that were more expensive and less available to them. So he wanted to help support his community in that way. And he also used it in his paintings as shown there. He was also quite the educator. Nowadays in progressive schools, we're all about like hands-on learning, project-based learning. He was already doing that. Again, sort of very contemporary education. And he built a mobile agricultural school where he went around to his community and taught them about best practices for farming so that they could grow the best crops to support themselves. And so he'd go around and try to engage with young people. He strongly believed in hands-on learning. He wanted students to learn common tasks uncommonly well. So not only was he an artist, he was a scientist, and as well as an educator. Before, again, sharing a little bit about my Bioart curriculum, Bioart, I'm not suggesting that this is a new innovation. It is not. In fact, Bioart is really reconnecting to our humanness and our connection with nature. One thing that I do with my students is I lead a book club that goes hand in hand with the Bioart project that I'm about to talk about. Braiding sweetgrass, I read the YA version, which is great if you have you know, a student in 7th to 12th grade. Actually, after each chapter, there are wonderful discussion questions or reflection questions that they can then ask themselves and reflect on their own connection with nature and their own practices of sustainability. And so I think it's super important when you're thinking about building the future of science education is to bring in a lot of diverse perspectives as to what science is. So we're not just talking about Western science practice that was sort of formulated back into the 1600s in the Royal Society, but we're also exploring other ways of knowing as well. And I feel like that is much more expansive, and we can understand more about the universe around us when we have multiple perspectives. All right, so now I'm going to be talking about Bioart and why it is so important and I'm so passionate about teaching it. So what is Bioart? Bioart is the use of organisms or biological processes to create some work of art. My teaching philosophy is rooted in the potential of Bioart to establish genuine connections between students and the natural world. I also just want to point out this beautiful piece of work done by my student. She grew different varieties of mushrooms through this tapestry with yarn that she dyed with various natural materials. So one thing that I do with my class is I get them trying to connect with their local environment. So we go out on a mushroom foray and we take specimens back to the classroom. And this is a really great way for students to learn that it is okay to touch mushrooms. That is a question I get a lot. They're always very concerned. I'm like, no, please touch them. And this is a moment where they talk about, they're super excited about, and it is just like a wonderful time to be in nature with students where they're actively searching for these hidden mushrooms in nature. So we do take some specimens back in the classroom. We can do a whole variety of things with them. We can do like fungal barcoding, identify species, as well as do some bioart with them. So up here we have some Ganoderma mushrooms. So these like grow a lot in LA, especially like in the end of summer in August and September. You might see these blooms around your street even. These were found like right outside of my school, and so we went out and collected them as we do every September. We bring them back into the classroom, we dry them out, and then one thing that you can do with them is if you blend them up, you can actually make mushroom paper out of them. So those brown rectangles there are some mushroom paper drying. Additionally, we can clone these wild mushrooms in the classroom as well. And so one thing that we can do is students are actively taking part in real mycological techniques here in the cloning process, so we can grow the mycelium and grow the, for example, we can grow them into mycomaterials, and this is some mycelium leather in the back here. If you don't mind, I can take questions towards the end. And then this is one of my favorite activities that I do with my students. So Dyer's polypore is this wonderful mushroom. It's more commonly found sort of north of us, but occasionally you'll find it near LA. And it's a mushroom that you can dye fabrics with. So it has like a range of colors from deep yellows to greens to browns, but it also has this really cool fluorescent pigment. So what you can do is you can extract that fluorescent pigment And then what I like to do with my students is you can do like fluorescent mushroom tattoos. So it is a semi-permanent ink. And so you can paint on it on your skin. It's non-toxic. You can even like dye your hair with it or paint your nails with it for a little bit more permanency. But it's a really fun thing to do with, especially like high school age students, because it's got this cool feature where it's invisible under white light, but it's only visible under black light. So they leave my classroom with like this sort of fun, secret, like tattoo, mushroom tattoo. And this also, handling these mushrooms in this way, painting them on ourselves, really lowers the barrier of that sort of mycophobia, that sort of fear of the other that we get when we're talking about mushrooms. And I've had students come up to me after this activity and like, wow, that's so cool. I had no idea that nature could produce things like this. And so it's just like a really sort of magical moment with the students here. Another thing that Bioart can offer is enhancing the scientific rigor. Oftentimes when I'm talking about bioart, because it has art in the name, people tend to think that, oh, this is not as like scientifically sound, or it's not as rigorous as a true like scientific class or curriculum. But I would argue that the opposite is true. And I'll give you several reasons for that. So first of all, the Bioart project that I do with my students is purely student-driven research. So they formulate testable hypotheses, they perform real experiments with unknown outcomes. They're moving through the entire scientific process to produce some sort of product. So here, students are moving through learning microbiological techniques, and they are inoculating fabric with bacteria. This is a really great one, and it also exemplifies the fact that in Bioart, you are collaborating with a living organism. And in order to successfully collaborate with a living organism, you need to know that organism on a deep level. You need to understand how do you work with that organism to get it to do what you would like it to do, or at least attempt to do what you want it to do. And so here, students are working with bacteria to dye fabric, and we can see that we get these really interesting patterns as the bacterial colonies are growing on the fabric. Bioart also promotes students iterating on their procedures and ideas, and also actively troubleshooting when things don't quite work out the way that they want. And that also occurs in, the research lab, when you're performing real experiments. When things aren't, working out the way you thought they were, you ask your friends, your colleagues, like, why do you think that might not have worked? You change your procedure a little bit. Maybe you look at a different procedure, and students are doing this in real time in the classroom as they're moving through their project. It also really strengthens and develops their time management skills. This is a semester-long project, so it's quite a lengthy project, and students are sort of forced to think out how they're going to manage their time. In the month of March, I have an entire month dedicated to in-class work time, where students have their individual projects, where they have to plan out, when am I going to be doing this particular experiment, and how long do I have to do that in to get the product that I want in time for the final event. Again, Bioart also enhances scientific rigor through the process of documentation. This is like a huge skill that they learn through this project. It's super important. Without documentation, you didn't do the work. That's what I tell my students. And so they take pictures, they take videos of their process along the way, and that helps emphasize the importance of the process, the scientific process, over the outcome, over that product. And so they can see how much work, how much effort they have put in troubleshooting, moving through those experiments in order to get to their final thing that they've created. And just a few examples, so sort of up top here to the left, I've got students who are documenting each day that they are extracting pigments from various mushrooms, preparing their yarn samples in order to dye that yarn. Over on the left side over here, or I guess your right side, students have documented the process of inoculating spawn jars all the way to using the mushrooms for mycomaterials. So it's quite an extensive process, and they're documenting and learning from each step of the way. The bottom left here is one of the highlights from last year. One of the students was working with my new favorite fungi, which is called hairy poop mold. And what high school student doesn't want to work with a fungus called hairy poop mold? It's this really awesome, gorgeous fungus that has creepy human-like hair qualities as it's growing from the auger. And, you know, if you learn a little bit about it, the reason it's called hairy poop mold is that, yeah, it does grow naturally on poop. But, you know, in the classroom, in laboratory conditions, it can just grow on regular auger. And it's like, again, it's safe to use, it's safe to touch, it's safe to alter and play around with. Imagine having like a chia pet instead with like hairy poop mold growing out of it. Amazing, that's what I want to see. So anyways, a student that had a blast working with this and documented everything with the name of the mold. So super fun working with students in that way. All right. And then Bioart also enhances the creativity within the scientific process. And ultimately, this leads to innovation. So I have a couple of examples of student work here. So I've got a mycelium hat that the students made. It's actually functional. You can wear it. It's pretty cool, a little top hat. And then we also have mycelium leather that students were able to prepare in the lab as well. Some other innovations that came out of this project was that we have this fluorescent pigment from this mushroom. We use fluorescent pigments in microscopy, in cell cultures all the time. What happens if you put this fluorescent pigment from a mushroom onto your cheek cells? Well, we can get a fluorescent stain of the cell membrane as well as the nucleus. So again, innovations even within various different fields. Last year I had a couple of students who were really interested in becoming engineers. They're actually in college right now for engineering. And what they wanted to do was they wanted to convert these 3D printers into bioprinters. And so instead of extruding PLA, they extrude bioplastics. And so they actually got it up and running and functional, and they bioprinted this little bioplastic heart. So again, Bioart also involves not only just like the field of biology, but it could be chemistry, it could be engineering. It's really cross-disciplinary. You can also do theoretical work in the realm of Bioart. And some of the theoretical work that I do with students is around biosensors. So biosensors are anything that reacts to like temperature, pH, salinity, glucose levels, and the theoretical part is like how might we use these in personal personal medical applications. So I had a student who's interested in making biosensor nail polish that would respond to pH changes. I also had students develop tattoo ink, and here they are practicing with that ink, seeing if it was a viable option on fake skin. And yes, I do have a tattoo kit in my high school classroom. Yes, I do get parental consent, and it's under my supervision at all times. They're only tattooing the fake skin, not each other. So this is also like trusting students in this process with you as well. Finally, Bioart allows for real, authentic output. So at Wildwood, we have this culminating event called Science Night. It's open to the public, and it's really just like a celebration of student science projects from middle school to high school. This year it's from 6:30 to 8 p.m. And this is on April 20th. And it's a really great way for students to share their work. So my students share their Bioart projects and talk to visitors about their process. They have their documentation that they're able to show the whole story of how they got there to their final product. And it's just like a really nice sort of final moment in this class. Additionally, last year I was collaborating with other faculty members at my school, from the arts department, from the math department, from our research institute, and we were able to provide students with an authentic experience and had an exhibition in Santa Monica. It was called Catalysts of Creativity, the STEM to STEAM exhibit. And I had students from my class present their Bioart pieces within this. And again, it was open to the public, and so people were able to come in, and students got to see this work outside of the classroom. And I feel like that was really important for them to see that their work was meaningful even without, being within the classroom, being assessed by me. And then additionally, I submitted a couple of student pieces, namely the mycelium leather and mushroom paper, to the Craft Contemporary exhibit, Materials Acts. This was part of the PST Art Initiative around Southern California. And we were the only high school that had samples on display here. So again, it was really powerful for these students to see that the work that they did in the high school classroom actually meant something to other people outside of their school. Finally, so I've been talking a lot about my advanced biology course. I also teach a neuroscience elective, and I've been trying to think about how do I incorporate Bioart into neuroscience. Now, neuroscience is a very abstract sort of thing talking to about when I'm trying to teach high school students. I have a background in neuroscience, so I developed this class at Wildwood. I thought it would be like super fun, and it really is. And what I've done in order to try to ground the more abstract concepts in neuroscience is to use electronics. So I get students to learn the basics of circuitry, learn the basics of coding through using Arduinos. And last year I presented their work at the LA Maker Fair. And this is the class project video here. They worked together to build a touch sensory system, and it does work. You can touch the touch sensory neuron, and then it transmits signals from neuron to neuron all the way down to the motor output, which is that spinning wheel. So this really helps students understand how do neurons actually talk to each other through electrical signaling in a more sort of grounded visual form. So bioelectronics is sort of an outreach or a branch from my Bioart curriculum. So with the remainder of time, I'm just going to show you a few examples of student work. So I have two main paths in my Bioart curriculum. The first one is biomaterials. Sort of under biomaterials, there's several different paths that students can take. So the first one is bio-leathers. So that's something like mycelium leather, mushroom paper, microbial leather. Here's examples of student work. So with the mushroom paper, you can see that students are able to actually like write on it. With the mycelium leather, again, this piece was actually the one that was displayed at Craft Contemporary. The microbial or kombucha leather. The kombucha leather is essentially just like the SCOBY that's dried. So instead of like making kombucha and like discarding the SCOBY, you can keep it, you can dry it, make this like creepy looking leather. You can also dye it with natural dyes to make it less like human flesh-like. But anyways, these are some really cool pieces that came out from my class last year. Another component under biomaterials is biocomposites. These are things that are made of one or more material or two or more materials. So we have mycomaterials and plant-based materials under this umbrella. A couple of examples, we've got the mycelium bowl, as well as the student who's really interested in trying to grow mushrooms in like unconventional ways. We tried stuffed animals. We also grew mushrooms on a book as well. I've got those on display over there. Plant-based materials, students were interested in growing plants out of fabric. So here they're investigating how do we grow plants out of like cotton, as well as we were trying to think about ways of how we could grow grass roots into a sort of woven fabric. And then the last one under biomaterials is bioplastics. These are algae-based plastics or bio-yarn. So for bio-yarn, it's just like these sort of long string of bioplastic essentially. And you can use that in a variety of different ways. Some ways that students have used this is by like through crocheting or using it as like string for their bio-jewelry. So they made the jewelry components out of bioplastic and then the string was the bio-yarn itself. Other examples, so bioplastics, we had a student that made a bioplastic cup, a drinking cup, and then this student over here, through many iterations, it was quite impressive the amount of troubleshooting that she had to do. She successfully got this like rubber like bioplastic that would inflate and created a bioplastic balloon. I wish she took a video of it working, but I assure you it did work. It was very, very cool. Sort of on the left here, you can see multiple different recipes that she used in order to get the right flexibility and consistency that she wanted her balloon to have. The second path in the Bioart project is biochromes, and so this can be multiple different organisms. We got fungi, plants, bacteria. So bacterial-based dyes, again, you can inoculate fabric and grow beautiful soil bacteria on this fabric to get really vibrant reds, purples, blues, depending on like pH. So a student like created like a crop top t-shirt for like some fashion design there. Plant-based dyes. Over the years, I've had students use like food waste products like avocado skins to things like marigolds to get like bright oranges and tie-dye some shirts with that. Again, like reusing old shirts and sort of upcycling them. For fungal-based pigments, students have used mushroom pigments in order to create watercolors and other paints and inks, and they also have dyed yarn with mushroom pigments. I've got those two little cute bears over there on the table with me as well. It was a super awesome project by Madison. So this branching out of Bioart, I'm also currently in the exploration phase of bioelectronics. So here is the biodata sonification device made by a student. That's the sound making device over there that was connected to the slime molds. So it's detecting electrical activity. All living organisms produce electrical activity, and so you can convert that into light or sound. And again, it sort of like grounds the students into like that electrical signals are a real phenomenon that you can actually measure. This was another project last year where a student, you can record your electrical heartbeat on your finger, and then it's attached to a motor, and that motor moves, and if you attach like some pencil lead to that, you can draw your heartbeat on a piece of paper. And then I just threw this in here, so I'm developing other ways to integrate bioelectronics into my neuroscience class, and this had mushrooms involved, so I was like, I gotta show you all. But the idea behind this concept is that it would connect to EEGs, so it could record brainwaves and then alter those electrical signals into different colors and LEDs. Again, trying to get students to really visualize the sort of invisible abstract electrical signals that are happening in their bodies. So these are some student testimonials from the Bioart projects over the years. So I'll read them out. I really enjoyed the Bioart project because it felt very personal. The Bioart project was a great way to explore the intersection between science and art in a very independent way. As an artist, being able to see how two of my favorite subjects relate to each other felt very innovative and inspiring. So I wanted to give you all some things that you might want to explore at home. Some easy ways to try out the practice of bioart. So spore prints, you already probably do that here, but that's a great way to sort of explore the different colors that are found in nature. Sort of along lines with that, natural pigments and dyes. If you really kind of put anything in a pot of hot water, you can extract some sort of like pigment out of it and see if maybe it doesn't have pigment, maybe it does. That's a way of sort of like exploring that realm. Sun prints or cyanotypes is another really great way to sort of see the shapes, the diversity of different ways that organisms have grown in the natural world. And then my favorite is nature journaling. This is a practice that I've been trying to keep up, but I think it's really important, especially introducing this concept to people at a young age. And this is for everyone. Everyone can make marks on paper. You do not need to consider yourself like an artist. It is simply the act of observing and then taking that observation and then putting that on a piece of paper. And no matter your artistic skill, you are observing some component of the thing that you're drawing. Maybe it was how the flower leaves were ovals. And that is actually strengthening your observational skills, which is foundational in the scientific process. I am also really excited to share that I'm hosting a workshop at Wildwood. This is open for educators and makers. And if you are in that field and you're interested in learning sort of a deep dive of bioplastics, I'll be exploring a variety of different recipes, bio-yarn, algae-based plastics, even like food waste leather. I'll also be showing how we can do bioprinting as well as doing soft robotics. So again, connecting back to the bioelectronics component there. How do we integrate bioart with the bioelectronics? So maybe you can imagine you have electrodes connected to you that can detect how you are breathing, and then perhaps that controls a air pump into like a flexible bioplastic balloon that expands and contracts as you are breathing. So there's a variety of different possibilities there, but I'm really super excited to offer this. And then I want to close out with some quotes by Jane Goodall. I started this talk talking about hope. I think Jane Goodall is sort of, has really shown us all that is a really important and impactful thing. And so just a couple of her quotes. I do have reasons for hope. Our clever brains, the resilience of nature, and the indomitable human spirit, and above all, the commitment of young people when they're empowered to take action. To reconnect with nature is key if we want to save the planet. So, thank you so much. I just want to give a shout out to my students. This work would not exist without them. Their willingness to experiment, to fail, to question, create, really makes this process quite meaningful. And I just want to thank them for sort of like trusting me through this process and really bringing it to life. And then here's my contact information. Just a heads up, my Bioart lab Instagram is just on the ground taking off, so I realize that there's like 8 pictures and like 3 people following me, but you gotta start somewhere. So I really appreciate the follow if you'd like to come along with me on that journey. And feel free to email me with anything. I'd love to be of help, and thank you very much. At this point, I'll take some questions.
Use the sterile area of Bunsen burner for sterile work?
I used to, and now I actually got sort of like a sterile fan that I use instead. But Bunsen burners are really like a pretty easy way if you don't really have access to like alternatives to create like a sterile area for sure.
Like what would you say the like contamination rate is versus like the laminar flow hood?
Well, the laminar flow hood is like the golden standard. So if you have access to that, like that's awesome. But in sort of like a makeshift lab space or at home, flame goes a long way. I say if you are really knowledgeable about the aseptic technique, like keeping everything clean, like having gloves, not breathing into your workspace and working pretty close to the flame, I'd say that you get like a pretty high rate of like not contaminating your work.
Thank you.
Do your students ever surprise you with like something that they come up with and you're like, wow, I never thought of that?
Every day. Yeah, absolutely. I think the balloon one was like a really prime example. I also had some students a few years back that made like bio slime, which was like such a cool concept. They have a lot of fun messing around with that. But every year they come in, they try new things. A lot of the student samples over here are them just like innovating off of like procedures they either found online or off of previous classes. So it's always cool to see like, I had no idea what they're gonna come up with like this year. So yeah, it's cool. Yeah, Sam.
It just seems like it's never been a more difficult time to be a K-12 teacher, particularly in the United States. I'm just kind of curious of how you don't feel burned out, and just maybe I'm very inspired by how optimistic you are about this, and it's a really hard job, and I don't really see like a lot of people with PhDs who are putting this much into their curriculums. Like, what are your just thoughts about that? It just seems like really hard to put this much work into this sort of job. It seems like a really hard time for teenagers to be working with them in this time.
Yeah, it is challenging, I gotta say, and it's really great to, again, I have the privilege of working in a private school that has a lot of resources available to me. I am grateful about the amount of support I receive. I design my own curriculum. They have a lot of trust in that, and that I'm a professional in my job. I'm an expert at what I do, and so I'm allowed to be creative in my curriculum. I change it up like every year. Bioart is just like a part of my overarching curriculum, and I do try to keep it new and fresh, not only for my students, but for me as well. I try to dive into things that I'm personally interested in because I feel like that helps with students engage, and you know, if you're interested in something, they're gonna be interested in as well. And I don't know, I feel like the optimism, this is something that I truly enjoy. And I think that I really enjoyed being an educator. I really enjoy working with this particular like age group of like 11th and 12th graders, and generally just like high school students as well. And I think that working with young people, it's not only like I'm hopefully inspiring them to like pursue biology or the sciences, but actually get a lot of inspiration from them as well. An example of that is like, I'm like super into like circuitry and electronics right now, and that really stemmed from like a couple of students last year who were really into robotics. And I was like, oh, that's sick, I want to try that out. And so, I don't know, I think I'm just like really lucky with like the school that I work at, the group of students that I get to teach, and I honestly like, this is what I want to be doing. I want to be doing Bioart, and that gives me joy. And, I don't know if that answered your question at all.
Do you see any greater increase in like interest and popularity of mushrooms in popular culture and things from like lion's mane and reishi and those kind of medicinal effects?
Well, I think that living in LA, we live in a certain kind of bubble, and I think that, I mean, even in California, I think that there has been an increase in interest in mycology and like things like, you know, medicinal mushrooms like lion's mane, reishi. I often wonder how my curriculum would be perceived outside of California and other states. And I mean, it's concerning how mycology is perceived, I think, outside of California, which is overall pretty accepted.
I saw a lot of the projects had QR codes attached to them. Could you explain that?
For sure. So that was from, I was taking a program called Fab Academy, which is a program that stems out of MIT Fab Lab. And in the final project, I was developing the sort of early stages of like the Bioart project piece, and part of that was creating like a living bio-library. Some of these samples here, I'm actively like storing student work in hopes of creating like a materials library so that each year students can build off of previous students' work. And in this project, they also create a website, and the QR code connects to their website. And I attached the QR code onto like these jars that were initially housing the materials, and I was like, jars don't really work very well. And so I'm moving onto this. I just haven't really touched the QR codes. I haven't really thought about how to integrate them. But I do use them at like science night, where like people can like click on the QR codes and like look at the website if the student isn't at their table. But yeah, that's sort of the idea behind it.
Have you, with your recent interest in circuitry, have you thought about using micromaterials or biomaterials as a substrate for PCB?
Oh, that sounds awesome. I would love to explore that. I definitely have been thinking about like, so for bioplastics, incorporating like conductive material within that would be really cool. I tried to like incorporate for one of the projects like charcoal, which is supposed to be conductive, it didn't work out that well. But you could potentially work with like that sort of biomaterial in a conductive way to create like circuits.
I do have a friend who developed developed a method to deposit copper onto PLA without damaging the PLA, so I could talk with you about that
Yeah.
after.
Absolutely, I'd love to hear it, for sure.
Hi, thank you for your presentation. It's very inspiring. I understood that the school, the courses, classes, curriculum you made kind of owe to the environment of the private school and, you know, very supportive and meets the challenge. If one wants to replicate that in the public school, I'm sure it's difficult, but do you have any angles that you would pursue? Because not everything is not going to be possible, but what would be the direction that could kind of like scale to the level of public school?
Yeah, scaling to the public school, like that's the dream. And I think it's really challenging with how our educational system works currently, especially science education is very tightly structured,
Sure.
with very little wiggle room for teachers to make their own curriculum. I think this also stems from a lack of trust of teachers as professionals in their own right, and I think it's very like a systemic problem within public education. And I think, you know, in, you know, the meantime, I think that there are some things that we can do. I think informal education that supplements public education. So for example, after school programs or like mobile school programs, like I'm thinking like mobile maker spaces or things like that that you can bring to the students and engage them in this work in that way. I think those are powerful tools. I also strongly believe in open source as education. I'm currently working on my website right now, in which all of this, all my lessons plans will be open source. And I think that accessibility to education at all levels is like super important. And while I don't think I can personally change like public science education with like Bioart at the current moment, like this is I think a step in like rethinking about like how do we get this to those teachers? Like how do we say that this is like a really great way to learn? Like project-based learning, doing science is so important. Yeah, I feel literally
Yeah, I was thinking it's easier to sell as a art course. You know, art course is more driven by creativity and curriculum language.
Absolutely,
Yeah,
that's
that. And the science is as like kind of agenda, hidden agenda kind of thing.
Yeah, I mean, I don't know too much about like public school, like art curriculum or like system, but like, I don't know, whatever works would be awesome. Yeah, for sure.
Thank you.
This is a YouTube question that came in, or comment, I'm just giving you a heads up. Congratulations on a Fastic Talk AM. Wildwood and its students and faculty are beyond lucky to have you. I wrote pages of questions and inspirations to ask you about at work tomorrow, Kate. I just thought that was nice.
Appreciate that, thank you.
You're welcome.
Great, I think that's all the questions. I'll be over here for a little bit if you'd like to chat, but thank you so much.